Well, today we're going to talk about complement deficiency syndromes, specifically CH50, and there'll be a bit of lab stuff involved. I know when I was studying for my physician's exam, I really wanted to understand the laboratory side. Maybe that's just me, but not just immunology, but also thyroid assays, because if I could sort of understand the assays and how they measured it, it sort of made more sense to know what they were measuring and how to put that into clinical practice. But I'll take this opportunity to explain the classical complement pathway and why we measure CH50. Now, this was a question over on my Instagram feed, a reel talking about why we order CH50, and this is the companion to that, specifically the details and a little bit more information about why we do that. Well, of course, this is sort of revision for the complement section in the Primer of Clinical Immunobiology. So if you want to read along, then flick open to this page, and this is what we'll be reviewing today. Okay, so let's, without further ado, head straight to the complement deficiency syndromes in a nutshell. Obviously, you could spend a week talking about complement deficiency syndromes. It'd be a very long week, but nevertheless, there's plenty out there. But you don't have time for that, and you don't need to. All you need to remember is the nuts and bolts of complement deficiency, and they are summarised here just for your revision. They're in the book, they're in the lecture series. But the classical pathway, C1, C2, and C4, deficiency of those are associated with autoimmune disorders like immune complex disorders, but also, as you could imagine, pyogenic infections where the classical complement pathway is implicated, specifically if you have antibodies to those types of bacteria. Now, you remember that all three pathways of complement converge on C3 to C3b step. And so if you're missing C3, obviously there's a lot that's going to be going wrong. You suffer from severe pyogenic infections as well as all the immune complex type conditions that we associate with the early components of the classical pathway, specifically when they're deficient. So the other things are related to Neisseria. So the terminal components, the membrane attack complex, if it's deficient, manifests as Neisserial infections. Similarly with the alternate pathway deficiency, propertin and factor D also are important in protection from Neisseria. Interestingly, and I make this point a fair bit, you're lacking this very significant part of the complement cascade, yet the only non-redundant infection that you suffer from here is Neisseria. So that's sort of interesting about how many layers the immune system has to compensate when something is deficient, but not enough to protect from Neisseria. Okay, so here's the whole classical complement pathway in a nutshell. You should know this, I hope, by now. Starting with C1, C1QRS, interacting with immunoglobulin, recruiting C4, converting it to C4b, converting C2 to C2b here, and C4b2b is a C3 convertase. This converts C3 to C3b. C3b can then interact with the C4b2b to make a C5 convertase. C5 is converted to C5b, and that then activates 6, 7, 8, and 9, eventually forming the membrane attack complex. So that whole pathway is what is being measured in the CH50. Okay, so let's sort of just work through how this is done, just really to illustrate what's going on in that previous slide. So basically you take sheep red cells, because they're fairly easy to get hold of, and you add an antibody to it, an anti-sheep red blood cell antibody. It doesn't really matter where that antibody or what species that antibody comes from, as long as it can activate complement, human complement. And then you add your test serum. So now we have a red cell coated with antibody. Nothing really is going to happen in the test tube until you add complement, When you do, that whole pathway goes to completion. Holes are drilled in the target cell membrane by the membrane attack complex. The red cells are released, and the amount of free hemoglobin can be measured on a spectrophotometer. The more complement, the more lysis of these red cells, the more colour on a spectrophotometer. You plot that down and you find the dilution that gives you 50% lysis of all the red cells that you started with, and that is the CH50, and that's where the name comes from. Well, you would go mad if you're a laboratory scientist doing that every day of your life, so we have come up with better ways of measuring this. One of which, and in very common usage, is haemolysis in a gel, where you basically do the same sort of thing, take sensitized red cells, in other words, red cells coated with antibodies, stick them in a gel, put them in the freezer or fridge, and then you add patient serum, and the serum will diffuse out through the gel. Where there's complement, there will be lysis, and so it goes up, using up all the complement until it gets as far as it can go, and the distance or the area that has been lysed reflects the amount of haemolytic complement in that specimen. So you then compare with standards to try and work out what the number is, and that will give you a clue. Obviously, if you look at these ones, you can see where lysis is occurring, and you can see where lysis is not occurring, and those ones would be perhaps of concern. Just as an aside, remember that the most common reason for an abnormal CH50 is that the specimen deteriorated in transit. If you heat complement, it loses its activity very quickly in a test tube after it's been bled. So just remember, before you go announcing that you found a patient with complement deficiency, remember that make sure that they have had the collection done really carefully. All right, are there better ways of CH50? And we'll come back to that in a moment, but first I just wanted to mention the alternate hemolytic pathway. Now, this is rarely done, and I'll show you circumstances where it might be done, but not many labs will do this compared to CH50, which is a fairly common, for some reason, assay. Now, you can ignore this side a little bit, because basically you don't use sensitized red cells, you use guinea pig red cells. Well, you remember that the alternate pathway is activated by a foreign surface, and it turns out that guinea pig red cells is a very good foreign surface to use in this circumstance. I don't know who first worked that out, but suffice it to say that it was worked out and you can use it. So here you don't have to add antibody, you just use guinea pig red cells and you add in complement, and that will kill the target proportional to the amount of alternate pathway activity. You can turn off the classical pathway by using EGTA to remove all the calcium, so that stops the classical pathway working, and at the same time add magnesium, which is optimal for the alternate pathway, so you can just measure one pathway in isolation. Of course, you're not really dealing with the classical pathway here because there's no antibody to be added, but just as an extra safeguard to make sure that everything is in order. Okay, I mentioned other ways, more automated ways of measuring CH50, and these are all sort of interesting, and obviously you don't need to know about it unless you go into a laboratory, but it's important that you understand it because it illustrates so much about the complement pathway as as a biological system. So here we use an ELISA on a plate, and we add an activator, and we measure. Well, once the activator is on, the rest of that pathway will assemble as we have outlined, and then you can come in with an antibody to the final component, and that antibody will only pick up or detect anything if all those components are intact. So it's a fairly cute assay, nice way of working, and you can change the activator to measure different pathways if you so wished. So you can change from IgM to mannan to measure the lectin pathway. I don't know why you'd want to do that, quite frankly, but it's done, has been worked up. Or you can use lipopolysaccharide as an activator to the alternate pathway in the same sort of principle. The one I came across most recently is a liposomal assay, and the principles are very much the same. So let me just work through it with you. You have a liposome and you put an enzyme, G6PD dehydrogenase, in the middle of the lysosome, and you put dinitrophenol as an antigen on the surface, and you come in with an antibody to that. Remember, it's just really much the same as sensitized red cells in a nice little capsule. You then come in with the patient's serum, and this will contain C1 right through to C9. That will then bind to that complex. The liposome will then be lysed by the membrane attack complex, release the G6PD, which will then convert G6P with NAD to NADH, and you can measure the amount liberated, again by spectrophotometer. So all these variations on a theme. This one's nicely automated, so it's quite attractive. Okay, so once you've done those complement assays, how do you interpret them? Well, it helps you by combining the results, narrow down where the deficiency lies. So if you have a deficiency of the common pathway from C3 through to C9, you can understand that this will affect both the classical and the alternate pathways. If you just have a problem with the alternate pathway and your classical pathway is working, in other words, the CH50 is normal, then it narrows it down to a few small components in the alternate pathway, including properdin. And if only your CH50 is down and your alternate pathway is normal, that obviously means that the classical pathway is deficient. So that's all I wanted to say about CH50. I've used this as an opportunity to revise the classical complement pathway in case you had forgotten it. I wanted to revise why we order CH50 and maybe AH50 in those circumstances that I alluded to. But basically it starts with a patient who you suspect has a complement deficiency syndrome, and that's usually manifest in the manner that I've outlined, specifically propensity to infection and sometimes immune complex disease for those early parts of the classical pathway. And then, but if you come back with a low CH50 in someone who you suspect may have complement deficiency, you can then go on to do further testing, and I outline what that might be in the primer, but basically it's to do other things like AH50, measure the individual components separately, or perhaps genetic testing once you've narrowed down where the defect lies. All right, so I'll finish up there just to remind you of all the learning resources that you have available to you. I will post another lecture in the near future. I hope that was helpful. Feel free to leave feedback, like, dislike, email me, whatever you think may be helpful to you. I will catch you next time.